Diabetes is a human disease. People who suffer from diabetes have an imbalance of the hormone insulin and cannot regulate their blood sugar. Diabetes can be fatal if not treated and controlled, especially type 1 diabetes, that is the childhood diabetes. Treatment of diabetes is important and has only been developed since about 1920. It was first had to be discovered that insulin was the cause, or lack of insulin. And then people with diabetes needed insulin. So where were they going to get insulin from? Insulin was originally purified from cows, and this is an animal-derived insulin. And now scientists have genetically engineered bacterial cells to produce human insulin. Instead of a cow or a human making the insulin, bacteria were genetically engineered to produce the human insulin. And no one has an issue with taking advantage or killing bacterial cells. So it was engineering of the genetics of the genes. Engineering the DNA was how this came about. The genetic code for human insulin, that strip of DNA, was inserted into the bacterial DNA. So a bacterial cell has its own genetic code, its own DNA, and this now is combined, the human DNA with the bacterial DNA. That's called recombinant DNA when you have DNA from two different species, the human plus the bacteria. This DNA then gets inserted into the bacterial cell, and the bacteria goes on and lives its life, and then as this bacteria grows, it is producing this insulin that it's not using, break down the bacterial cells and recover the insulin, and then use that insulin as a medication for humans. So how DNA can be combined from two different organisms is because the DNA itself has the same components, the same monomers for bacteria DNA as human DNA. What you're looking at here is a representation of all the genes of one human organism or one single person. This is called a genome. That's all the genes that code for every protein in a person. There are 23 pairs of chromosomes, 22 plus this is the sex chromosome. So there's a total of 46 chromosomes. Each horizontal dark bar that's shown in here, and there's a lot of dark bars, each horizontal dark bar is a cluster of genes. So there are a lot of genes in a genome, as you would expect, because there are a lot of traits and a lot of proteins and functions to code for a single person. So humans have a genome, bacteria have a genome, and each organism has its genome, which is the guidebook of how to basically grow and reproduce that entire organism. So if you had a guidebook of how to make dinner, you would have different chapters. You would have the chapter on salad, and then you'd have the chapter on the main dish, and then the chapter on the side dishes. Each one of those chapters would then have recipes, and so that would be broken down, and that's what happens with the genome. This is all the information, and that information is broken down into portions that are these are different chromosomes, and then within the chromosomes there are genes. Those are the individual instructions. Those are the basic units of heredity. The individual instructions that all put together are the giant guidebook of how to make one organism or one human in this human genome. One gene is the information to form one protein, which is usually going to be representing one trait or one function. This is something that is heritable. DNA can be passed down from the parent organism to its offspring organism, and that's inheritance. Here's what's interesting, though, about inheritance. If you change one gene, you can change that protein, or you can change that one inheritable trait. It's such as the example of switching the color of corn from white to yellow. Changing one gene can change the color because what you're changing is the protein that causes this to be white or yellow. So small changes within a gene are usually not enough to make a cell produce a new chemical like insulin, but you need a whole new set of instructions inserted into a genome. And so that's what's behind recombinant DNA. Taking a gene from one organism or a series of genes from one organism or even genes from different organisms and then inserting them into bacteria to get this bacteria to make a protein that then you want to use